floor. So with that, please join me in welcoming Joel Cracraft to deliver his lecture on Amazonia, Reconstructing the History of Earth's Most Diverse Biota. Dr. Cracraft.
All right. Thank you. Can you hear? Oh, you can't hear me. Right. Well, this is an honor, and being elected to the Philosophical Society is especially so. It came as a big, big surprise and a very pleasant one. Can we turn this down just a little bit to? No, this was turned. Now it's off. Okay, I'm sorry. I turned it back on. Well. Okay. All right. So it's a real pleasure to be here. And I want to thank Larry for inviting me and for the wonderful hospitality we had at dinner. Tonight I want to talk to you about Amazonia. And this is a talk that is to let you know about a big, big effort that many of us are putting in to understanding the history of Amazonia, particularly over the last 5 or 6 million years. In other words, the Pleistocene history. And it's being supported by the National Science Foundation and NASA here in the United States, and the São Paulo Foundation for Science in São Paulo, Brazil. And it's also a core project of Biogenesis, which is a project part of the organization of Diversitas, which is the international science consortium. Amazonia is vast, and it's diverse, and it's very important for all of us. It covers 9 nations. It's 7 million square kilometers. There's a huge amount of taxonomic ecological diversity in Amazonia. And it has had a very complex and deep environmental history. But it's globally important for all of us because the total forest biomass is about 100 billion tons of carbon. So it's a major source of carbon in the atmosphere. It's a major sink for carbon in the atmosphere. And 20% of atmospheric O2 flows through Amazonia. As 10% of global terrestrial net primary productivity, it covers 15 to 20% of global fresh water flows. There are about 25 million people as of a couple years ago in Amazonia, and forest loss is 20%, and it continues to increase. Amazonia is all this area, we kind of define Amazonia as all this area in green, which is mostly lowland forest, but some in mid-montane, low-montane, and montane areas here, and various intercalations of rainforest through riverines up into this very dry area of Brazil. Amazonia, as I'll come back and mention again, can be divided up into many areas of endemism. Areas of endemism being where there are multiple species found there and nowhere else. And these are kind of iconic vertebrate areas of endemism. They don't necessarily fit all groups of organisms, but things like birds and primates and some other groups, there's a lot of diversity and endemics here. In other words, a lot of species found in this area but nowhere else. So one of our problems here is how is all this genetic diversity, if you think about millions of species being in Amazonia, how is that genetic, taxonomic, and ecological diversity distributed within Amazonia? That's a big, big question. What has been the evolutionary history of the Amazonian biota, and how is that diversity generated? This is a question that has been on people's minds for over 100 years. There's a huge amount of diversity. How did it come to be? And another way of looking at it is how has the environment and the diversity evolved together? It's a very complex geological environment in Amazonia, and there's been a lot of controversy over how the Amazonian terra firma forest and the riverine systems were all originated, and we're going to be talking a fair amount about that. And then another interesting question is, how is this co-evolutionary system? You know, Earth and the biota evolving together, how has this contributed to the global impacts on Earth systems? And I'll briefly mention this. So understanding Amazonia is a question of how does ecosystems at various different scales assemble themselves over time. And by assemble, I mean how does the taxonomic diversity and the ecological diversity arise over time. And so this question, the answers to this kind of question are a matter of spatial temporal scale, looking at things at very short temporal scales and things at very long temporal scales. And so we can see Amazonia in a kind of context of the whole continent, because things in Amazonia are related to how the continental biota assembled itself over time. And within a continent is the regional biota. And we can say the regional biota is the Amazonian biota. There's a biota of major areas in South America that are Andean, some that are in these dry areas of South America. But we'll focus on this regional biota, and then we can look at landscape scale areas of endemism, like I showed you, within this big local, this big regional biota, and at the local community level. Major questions within biology are, how do each of these smaller scales, how do the biotas within those scales relate? One of the things they don't do is they often just go in and study the local community. But the local community is embedded in a landscape. The landscape is embedded in the regional biota, and the regional biota is embedded within all of South America. And so to get more specific, we can see all of South America, the biota, and within it is the area of Amazonia. Within it is this area of endemism, and within it is this local community of Coxacashu. So the birds that are present there have a history, and that history is related to the history of this area of endemism, and the birds there is related to the history of all of Amazonia, and the birds in Amazonia are related to how South America as a whole got its avifauna. Now, I want to go through a few slides to sort of, with a series of cartoons, to show you how we think about solving some of those problems of history. And one way of looking at it is all of these ecological assemblages are historically structured. And we can tease apart that structure by understanding the phylogenetic relationships of the species within those areas. So we can start, for instance, at time 1 with this ancestral biota, and you could pick this at any time span and run that clock out. It really doesn't really matter. You just go from one time to another. But we have this ancestral biota, and let's say we have a local biota that we're interested in also. Well, barriers can arise, and when barriers arise, they split up these descendant biotas. And this barrier could be a mountain range, it could be a river system that arises, it could be an ecological change that is a dry corridor between two rainforest areas. But we can reconstruct this, and I'll show you some real world examples. And during this time, species can be lost to extinction. But in these areas, species in isolation then can evolve. So the most common form of speciation is this allopatric. In other words, these two areas are divided, and the species within these biotas, by and large, are also isolated from one another. And over time, they will change into new species. They will evolve into new species. But then, sometimes these barriers go away, and biotas can come back together again. And we call that biotic dispersion, where biotas move across the landscape as long as the landscape, the climate, the topography and so forth allows them to do it. But biotas can come back together, and now you have a mixing of biota A with biota B. And then there could be a subsequent, another change. We call these vicariance events, from the word vicar, and they are, biotas are replaced, replaced by other biotas across space. So biotas can split apart, they can come back together again, they can split apart again. There's also historically unstructured biotas. So you can have a biota and there could be long distance dispersal across pre-existing barriers. And there can be isolation. So some species that evolve in this area could have come from outside. And it can be very idiosyncratic, and it's not historically structured by replicates of species within these biotas like it was in the previous slide. And here's a way we can look at it. So you have a regional biota, and you have a local assemblage within that biota. A barrier can arise, and now if there's a differentiation on both sides, you have two replicate patterns with sister species here and a sister species there, here, there. And if those species respond to that barrier in more or less the same way, there could be a replicate pattern. So you get this congruence, and it's searching for that congruence over time that allows us to reconstruct the history of these biotas. Now, there could be a second barrier that splits this region, and now we have the blue species. Its sister species is right there. The yellow species, its sister species is right there. Now we have a much more powerful statement, because now there's been multiple historical events that are shared by different groups of species. These could be different birds. They could be, one group could be birds, one group could be primates. It doesn't really matter, but the history of the environment is creating speciation patterns that are replicate. And the more that they are replicate, the more information you have about the history of the big areas that you're studying. And then the barrier could go away, and now we still have this pattern, this historical pattern, but now when we look at the species, it's a little bit different. You have to understand the history of these in order to interpret that maybe a barrier disappeared. And there are ways to geologically investigate whether barriers disappear. Now, if we went down to the local level, we might see a local community with these two species. Well, their history is historical, so the fact that those two species are in that area is also a result of history. And the same thing here. And then here, gets a little bit more confusing, but if you know this history, then you have an idea that the barrier disappeared, and these species now commingle even in local communities. So this method, in general, in principle, allows you to understand the history of local areas, the history of bigger areas, and the history still of bigger areas, because you can expand out your analysis. So the first principles, I think, of looking at assembly, no matter at what spatial scale you're looking at, is that speciation is predominantly allopatric. In other words, it's controlled by barriers and isolation. Everybody, I think, in evolutionary biology understands that this is the predominant mode of how we originate new species. And these barriers are a function of Earth's history. And thus, geological processes, tectonics, geomorphological processes, climate change, they're all related to one another. Tectonics controls climate, tectonics controls geomorphology in various ways. And so if we see these factors as controlling allopatry, in other words, they're creating the barriers that creates allopatry. Both vicariance and long-distance dispersal can create allopatric, and they can lead to speciation. But it's really vicariance that leaves these historical traces behind that we can reconstruct using phylogenetic analysis. And causally linking biotic history with Earth history depends on discovering congruence in those two histories. And that congruence is largely spatial, and it's also temporal. And it's getting the temporal part that is complicated for us. So if we look at assembly, we want to understand environmental history. We want to understand the spatial history of the areas themselves, ecological history, and species history. And for us those of us who are systematists, we really want to understand what species are there. In other words, what evolutionary units are out there in nature. How are they related? So we understand, try to reconstruct phylogenetic relationships. And then another thing that's actually fairly young in systematic evolutionary biology is trying to estimate a time tree. A lot of controversy in that, a lot of nuances in how to do it, but we're getting better at it. And if we can reconstruct time, that's very important for looking at geology, and it's very important for looking at phylogenetic history. And then we want to look at the spatial history. So I showed you those areas dividing up. Now, we don't necessarily know all of those, but by reconstructing the phylogenetic history of big groups of organisms, then we can start teasing apart the spatial history and how areas were separated or brought back together again and are involved in the isolation of these species. And then we want to look at paleo-environmental history. Now, the sad thing is that we biologists have not necessarily understood how we might go about doing it. Many of us biologists, we're very good at doing phylogenetics, and we can do historical biogeography, and we can do what called phylogeography. In other words, we densely sample species or groups of species in areas genetically, and we can understand a lot about the history of those populations and those species by looking at their genetic history, and I'll give you an example of that. And so we biologists are over here, and then the geologists are over there. We are not looking at them, they're not looking at us particularly. And they just go on and they study paleogeography, tectonics, paleoclimatology. And if we biologists pay much attention to them, we either ignore Earth history and just do our thing, or we read some of their papers if we can. And generally speaking, biologists will pick those papers that have the really neat cartoons about, continents going apart from one another. And the biologists don't understand that the geologists don't agree with one another. Biologists understand that they don't agree with one another, but they don't understand that the geologists disagree with one another. And you can't trust one geologist over another. You've got to look at the evidence from a geological point of view. And you just do all this and you create some sort of narrative, but it's not a very testable narrative often, and it doesn't integrate the two. And the key here is that we need a new paradigm for looking at these big, large-scale ecological assemblages. Things like the birds of North America, things like the birds or the frogs or the biota of Amazonia altogether. You need systematists to do the systematics and the phylogeny. You need historical biogeography. So the systematists will be doing these kinds of studies. Then there's population genetics, looking down at the micro level of populations and within species and within local areas. You want climatology and palynology and climate modeling. And you want landscape ecology, ecosystem function, biogeochemical flows, and all this geology. And we need all of those working together to look at this problem of Amazonian history. And a whole bunch of us have gotten together starting a year ago in this big Amazonian biodiversity project. And we're actually the first one, but another one was just funded through geosciences at NSF. And it's a really complimentary group of people. We've met with them at a meeting, and there's a lot of synergy and a lot of people interested in this. And this is the cast of characters. It's 17 institutions in Brazil, Argentina, Great Britain, Canada, and all these in the United States. And those of us in the United States are funded through NASA and NSF. Those in Brazil through the FAPESP program. And all these others are tagged on to our separate grants and attending meetings and collaborating. Now, this is a complex problem. It's if you're a program officer at some funding agency, you're looking at this and you're saying, "Oh my God, do these people actually know what they're doing?" Because this is just a little simple way of looking at it. I could have put one up here with all the data flows that we envision will happen, and it would have gotten a lot messier. So we're characterizing Amazonian biodiversity. We are doing a lot of georeferencing of specimens because knowing precisely where specimens are located allows us to do a lot of computational work on taking this specimen at this locality, it's identified as certain species, and we can look across all of those data, which will eventually end up being more than 1 million data points on Amazonia before we're through with it. And you can do an awful lot of really cool science about patterns of diversity, patterns of endemism that you couldn't do without those georeference. So we're accumulating a lot of georeference specimens, mostly birds, primates, and plants. And we're looking at a whole big databases of abiotic and biotic variables across the whole region. Then we're looking at the history of Amazonian taxonomic diversity, phylogeography, phylogeny, genetic diversity across the area as subsamples of the species we're looking at. So if we have lots and lots of samples of many species across Amazonia, then we can do detailed genetic and population demographic analyses. We also have palynologists, geologists, hard rock geologists, climatologists looking at the paleo-environments, the Neogene river dynamics, geochronology, and also people looking at the functional systems and biogeochemical flows over time. And we can integrate all of these kinds of studies. So we can integrate the evolutionary history with the paleogeography and ask really kind of cool questions that you couldn't ask by looking at those studies by themselves. We're very much interested in trying to understand the spatial history of diversity in the environment and scales of environmental change. So we want to look at how the system, the Earth system of Amazonia arose over time, and then what are the consequences of that for the global system. So I'm going to now switch to a couple of examples of how we're going to go about doing some of this. Again, it's early on in the history of this program, and believe me, trying to handle 30, 35 people at 17 institutions like herding cats. And I'm not a micromanager, and getting everybody on the same wavelength and working together is a challenge. It is definitely a challenge. But it is happening. It is happening. So we're going to look at these wonderful birds called trumpeters. They're related to cranes, and they go back deep in Amazonia. They have a history. They link up with cranes probably somewhere in the neighborhood of 35, 40 million years ago. But they are endemic to Amazonia itself. So somewhere over that long, long history, there must have been these trumpeters somewhere else in rainforest, but they love the lowland part of the forest. They walk around, pick up fruits. They do fly. They go up on perches at night, but they are mostly in the terra firma forest, and they're pretty amenable for people to have them as pets around villages out. But they show really amazing areas of endemic patterns. And so we published a paper on them last year in the Royal Society. And we are interested in these areas of endemism in Amazonia because within these trumpeters, there are differentiated evolutionary taxa, species, in each of these areas. So the idea is if we can reconstruct the phylogeny of these species, we can at least have a first-order hypothesis of how those areas are related to one another historically. If we can age it, those birds, then we have a prediction about the ages of those areas and when they split apart. Now, this is based on one group. I'll tell you right up front, it gives a wonderful pattern, and I'll show it to you, but it's not necessarily a pattern that's found in other groups. So there's a depth of time and separation of these areas. So let's look at the tree. We have a tree, and here are all of these areas of endemism, and they're color-coded. So Sophia crepitans is in this whole big area of Guyana, area of endemism. Their sister species is Ochoptera, which is found in the Negro area of endemism, tucked in here between the Solimois River and the Rio Negro. And then out here in the far western Amazonia, north of the Solimois River, is Nepensis, and it's the sister group of both of those together. Now, everything else here, so these are all north of the Amazon, these are all south of the Amazon. And Leucoptera, out here in the Inambari area of endemism, is a sister group of everything to the east. Viridis, here in Rondonia, is a sister species to all of those. And then these three divide up with Interjecta here being the sister group of Dextralis, and those in turn are the sister group of the species in Belen. So there's a very clear historic spatial pattern in history that allows us to hypothesize. Now, we dated this phylogeny in several different ways, two different independent estimates, using birds distantly related that have very good fossil records, and we can calibrate these time trees using the fossil record, and we come up with this phylogeny being only around 3 million, 2.5 to 3 million years. All of this diversification happened in here in just 3 million years, not much more than that. And this, we think, is robust, and we've been testing it in other ways. Now, we can actually, these birds are not, there's not a lot of fresh tissue in collections of these birds. There are a number of museum study skins, and we've extracted DNA from museum study skins. We'll soon be extracting DNA from study skins for next generation sequencing, so we'll get a lot more data for these critters. But we have done some genetics in areas where we have multiple individuals from fresh tissue, and it gives us an idea here of looking at Amazonia from a population perspective. And when we divide these taxa east to west, so here's crepitans here, and then here are combinations of these two areas out here. This area covers these areas right through here, and then all of these areas out west. So we were interested in looking at genetic variation from east to west. And what it tells us, or at least suggests, we want to get more data, that there's more genetic diversity here. So there's more different kinds of what we call haplotypes in the east, here and here, and fewer out west. And that tells us that these have been around longer. There's more haplotype diversity, and we can postulate from that these populations up in the north and in the south both began in the east and moved westward. Now that's another hypothesis. It tells us another hypothesis about the history of the areas. And again, we're doing this, we're replicating this over many different groups of organisms. Now, the cool thing about the approaches that we're taking is that this presents a challenge for paleogeographers and for geologists, because from this we can infer the relative ages, certainly, of the river systems, because these things are separated by these major rivers. And so we can make a hypothesis, and in this paper we did. And the idea was there are two big competing hypotheses for the origin of the Amazonian river systems and the hydrological systems. And we can use these birds to test those two hypotheses. So we created a model. And the model is that there were Pliocene wetlands out west, and the ancestral distributions of Sophia were both north and south of the Amazon River. This is in the Amazon basin. This is the Solimoes basin. And I'll come back to that. This is very feasible in a minute. You'll see that it doesn't sound right, but it actually will be right in a minute. So this is about 3 million, 2.7 million years ago, we postulate. There's an arch right here, the Purus Arch, that is a big underground, it's all below the surface, but it has kind of been uplifted because when the Andes rose up, it created misbalance around the Amazon basin and lifted up. There's a rebound effect that lifted up this arch. And the drainage system, this is very well known in Amazonia, the drainage systems out here in the Solimoes basin and in the Amazon basin have been separated at various times. So there's this arch. We postulate that that arch was broken through by the accumulation of water out here, and it finally came through around 2.7 probably million years. There's statistical error around these dates, so they're just, they're the kind of the mean of our analysis. But these birds were isolated by this incipient lower Amazon River, but they moved westward because as this wetlands drained, the terra firma forest began to populate that area out west. And you can grow rainforest in a couple hundred years. So the times we're dealing with are immense toward relative to the times it takes to grow these big kinds, deep ecosystems. So the ancestors move south of the Amazon, they move west. North of the Amazon, they move west. That's what the indication of the detailed genetics seems to suggest now. At 2 million to 2.1, you can't even measure that given the methods that we look at. What happened was the Rio Madeira arose and separated off Leucoptera from everything to the east. And then at around 1 million, you had the Rio Tapajos arise and the Rio Negro arise, or a paleo-Rio Negro, as I will point out to you. We think it was a paleo-river system, not in the current area where it is right now. And at around 1 million, then the real Rio Negro arose and separated the species here and the species here. And then eventually, much later on in the Pleistocene, these river systems, the Tocantins and the Xingu, the Tocantins right here, and the Xingu arose pretty much at the same time and then separated the species out here. So this is kind of like a bold hypothesis for the geologists to look at. Now, like I said, there are two big hypotheses for Amazonia. One is that this whole river system that we see today is a Miocene river system. So that by the end of the Miocene, 8-9 million years ago, toward the late Miocene, that river system was in place. Then there's another hypothesis that in fact, the Amazon River is only about 2.5 to 3 million years old. That the whole hydrological system there is young. These are two hypotheses that our results can test, because we can at least say that it's the young river system that seems to be more supported by the data from Sophia, from the Trumpeters, than the older version. And it goes like this. It's a really complicated. We've got geologists trying to understand it, but a number of our geologists have really looked. Here in this basin, in the Amazon basin, they don't find Miocene sediments from the Andes and out west. Here in Amazonia, you don't find sediments out here until the Pliocene, after the Miocene at least. But the idea is that we have lots of information that back in the Miocene, maybe even into the late Oligocene, there was a very, very large river called Lago Pebas, Lake Pebas. There's a lot of evidence for that. And then it disappeared around 10 million. And now the geologists start to divide. When this disappeared, the geologists would argue, well, that's when it broke through and went out to the Atlantic. But the other geologists say, oh, hold on. If you look at the stratigraphy and the geology of western Amazonia, there was still another lake system that came in. So after this lake system, there was a peneplain. In other words, the land all the way out in western Amazonia had more or less arrived at the same topological flatness, and there was no more undercutting. But at that time, there was another really major uplift of the Andes, and it started dumping a lot of water into the western Amazonia and started to create a lot of river channels. So this Lago Amazonas, which is the second kind of lake system, is sort of like the Pantanal today, where you have interbraided river systems, sometimes wet, sometimes dry, depending upon the rainy season. And Lake Pebas exited out this way and out this way, and Lago Amazonas probably did the same. So the hypothesis is that as water built up here, there's finally erosion across this barrier at around 2.5 to 3 million years. And we're getting consistent geological evidence here to say that not only are there no sediments, but the ages of rocks in here and the zircons that are in these rocks, these little kind of crystal things that you can actually pretty precisely date radiometrically, all of these are from these upland areas, which have been there for a very, very long time. So the ages of these rocks are found in sediments here, but the ages of these rocks, which are younger, are not found in here until later on when this thing broke through. And there's all other kinds of evidence. It indicates there's a major influx of sediment from drill holes there at around 2.5 million years. And the idea is there had to be an Amazon River carrying that. At various times in the Pliocene, late Miocene, this was moderately dry. It depended on the water that was being dumped here and the water that was being dumped there, but there were probable land connections across this drainage system because the river wasn't that large at various times. And remember, these river systems did not evolve until later in the Paleo-Pleistocene. So this water system was probably draining in different directions. At least that's the sort of indication. And I can give you some ideas here of how rapid these rivers can change. And one of the things that we're studying is paleo-river systems. So this is a topographic map of Amazonia. And this, as I explained there, you have this ancestral distribution, and a barrier arises, and it separates them. So if a barrier arises and separates them, that's vicariance. That's a vicariance event because this barrier is creating very similar things that replace one another. The Pope replacing one another. It goes way back. It's a term that's been created now for 30 or 40 years, and it's kind of idiosyncratic. I do agree with that. But it's allopatric speciation by a barrier arising within an area and separate them. Okay, so we're going to look at the Rio Negro and the Tocantins here. And this is one of the lowest areas. This is probably about the lowest area in Amazonia. And if you fly over it from Manaus this way, you see that this forest is very different. You can actually see it from the air. You can see river channels underneath the forest. It's very bizarre. Whereas if you fly around here, it's just this sort of flat forest. So there's something really cool going on here, and a couple of Brazilian geologists looked at it not so long ago. Here's the Rio Negro. Here's a paleo-river system. This is all covered over with rainforest. But you can use SRTM radar data. So they will haul this machine behind a plane and look at paleotopography, look at the topography through the forest. And when they do that, they see this big ancient river channel right here. It's no longer used. These other ones are rivers that are cutting across it. And when you do a profile across that area of topography, this is what it looks like. So there was a big ancient river valley from here to here, about 12 kilometers wide. And now the current rivers are cutting down through it. And you can see this river cutting down through it, that river cutting down through this big river channel. No, kilometers. Here, vertical? No, meters. Yeah, meters. And so there was a big system. And the only thing it really could have been is an ancient Rio Negro. And cores in the Rio Negro show that it's less than 1 million years old, right where it is right now off of Manaus. So one of the things we want to understand across Amazonia, these data are available, they just haven't been analyzed to ask the kinds of questions that we're sort of interested in. We want to construct these kinds of observations for all the big rivers to try to understand where were they in the last 1 or 2 million years, and go in with field geology and try to date those rivers. And they've done that along the Rio Negro, and there's consistent that this right there is very young, geologically speaking. We can go across the continent to the east and look at the Rio Tocantins. And we can see that this is if you look at a geological map, there's a huge swath of Paleo-Pleistocene sediments in this big ancient paleo valley. And the interpretation here is that in the Paleo-Pleistocene to maybe mid-Pleistocene, the Tocantins River drained this way and deposited all these sediments out in here. Then, in the late Pleistocene, probably by faulting, changed the river from this angle to that angle. And then subsequent to that, the Para River cut across that. So you know that this is younger than that. And this has been more or less dated, and this event in this Pleistocene to mid-Pleistocene, and the vicariance event of Sophia across that is around 700,000 years. So it's easy enough at least to postulate that it was the major shift of these rivers that happened really quite quickly, because if you can strike slip fault fairly rapidly in geological terms. Now, we can also extend these kinds of observations down to the genetic level. And we're just experimenting with this. And so I'm just going to show you some what will be or what could be when we look at it. If we look at the genetic structure of the species in Rondonia, we see that there's a lot of genetic structure. So this is mitochondrial DNA. These are the individuals we sampled. And these are the mutation steps in these genes that we studied. And you can see it's highly structured. Highly structured. Whereas overall here in this very, very large area, all of these things are only one step away. So there's two individuals one step away that share the same genetic structure. One individual here, three here, three here. But there's very little genetic structure compared to here. Now this tells us something. This tells us that this genetic structure took time to get like that. And hence, the environment was more stable over longer periods of time. Here, this suggests that there was a recent expansion of the population in this whole big area of endemism, and it hasn't had enough time to develop genetic structure. Now, these make two different predictions, and I'm working with a geophysicist climate modeler, and I say, now, what do we have to do to explain this geologically? And so, while we were at our meeting last year in Sao Paulo, she said, well, let's look at inverse vegetation climate modeling. You tell me what areas are stable and what areas are not stable environmentally, and I will tell you what parameter sets in these climate models can give us those estimates. And so she changed the climate, last glacial maximum climate, 5 degrees cooling. She cooled it down 5 degrees, 20% reduction in precipitation relative to today, and a reduction in atmospheric carbon dioxide. And she's able to predict with those parameters that this area is stable. It remained rainforest through the last glacial maximum. Whereas this area up here changed more to savanna, open area. And so therefore, if the Sophia up here expanded after that time, it's a younger time than what the Sophia in Rondonia has. So we can replicate this kind of analysis group after group after group, and there are other things published already in different groups that indicate that these areas of endemism that we can easily reconstruct actually have different local environmental histories. So this is going to be kind of cool, marrying, a real hardcore a geophysicist that does climate modeling. That's going to be kind of cool. Now, the implications of all of this that I've given you is that the climate models of the current world, the climate models of the Pliocene for the global vegetation, if the model that they're young, that the Amazon basin is young, then all those climate models can't be right. Because this system puts so much carbon and carbon sink, and if the wetlands out west were vast and no terra firma forest, then the methane budgets have to change. So we think that these kinds of studies have major implications for tweaking the paleoclimate models of the past And it gives us a window into how did Amazonia today come to be. And that's pretty important because if you understand how it comes to be, then you have a better refinement for saying where is it going to go under various climate parameter sets. So to sum up here, understanding the history of these systems is really complicated, and integration across disciplines matters an awful lot, it seems to me, for getting this all right. We need to know phylogenetic history, we need to know the time trees, we need to know the genetic systems of these species, but we can't really explain that until we get the geology right. And that means geologists and palynologists working together. And some of our palynologists are putting down new cores in areas that are going to give us really interesting information about the history over the last 5 or 6 million years at least. And so integration across disciplines with these large-scale projects is really necessary. So I want to thank all my collaborators. This is a cast of characters that got together in Sao Paulo, and we're getting together in Manaus in February again. And my funding sources, and I want to really thank my hosts. It's been a wonderful visit, and thank you very much.
Thank you. You have time for some questions?
Absolutely.
So if two mics going around, please raise your hand, stand, state your name and whether you're a member of the society or not. Let's start with William back there.
William Worley, I'm a member. How do you relate this to how the climate is shaped by the Amazonian region?
Well, we're going to do that by modeling. Our hope is to do enough history of the taxa to get an idea of the ages of the areas, what environments were there, and then we can use those as to modify the parameters of these vegetation models and the climate models and get them more refined. We have knowledge that there's dry areas in Amazonia. These forests can go away and be replaced by savanna. But the current climate models show this big swath of forests going from the south to the north. Yet our birds, and I could have shown you another example of this, but our birds are older than those postulated changes. And they require lowland, a rainforest, wet forest, and so those birds and a lot of other birds indicate that there couldn't have been a dry forest through there. But with these kinds of information, I think we can get at the locations of some of these dry areas in a much more refined way. And then looking at palynology, new cores, palynological cores, we can get a really clear history of what forest was what. There are very few cores in the Amazon basin. Most of them are on the periphery, and they show Pleistocene changes from forest to savanna and back. But down, the few cores in Amazonia itself seem to indicate through the last glacial maximum rainforest. But some of our group is drilling cores that actually go way deeper than the Pleistocene, or go deeper than the Pleistocene apparently. And once we get magnetostratigraphy on those, we'll be able to really pinpoint the ages and pinpoint what plants were there in those sediments. So it's kind of cool.
Let's get the gentleman there. Let's get on the other side. Okay, well.
Thank you. My name is Rudy Curator. I am a member of the society.
Can you move? Speak. Yeah, thank you.
My name is Rudy Curator,
This on?
and I'm a member of the society. You made a claim, as far as I know, unsubstantiated, that the Amazonia was the most diverse biota in the world. How does it compare to Africa, the Pacific Ocean, Indonesia, and other places with similar climate?
Yeah, that good question.
You have a measure.
Yeah. I mean, you're absolutely right. We don't know how many species are there. But if you look at the plants, you look at the animals that have been inventoried, Amazonia, which is huge, is way more diverse than Africa, which is broken up into these very isolated rainforest areas now. And the same thing with Southeast Asia, but that may not be true for all groups of organisms, for sure. So what I was referring to are vertebrates in general, but not any specific ones, because there are more different kinds of one kind of bird over in Africa than in South America. But there have been estimates based on what is already described, which is probably an order of magnitude less diversity than is actually there. And Amazonia, I think, is the most diverse, but of course it has the bigger rainforest area of any place in the world too. So that's where that kind of statement comes from, and I think by and large people agree with that general statement.
My name is Al Ehrlich. I'm a member, and if I might, I have two questions. One is simple enough. The winds in South America are prevailing westerlies as it is in North America, are they not?
Yeah, they come there, but also from the Atlantic too. You know, that's the other problem. The intertropical convergence zone moves up and down.
Because my problem was that in North America, the area just to the east of the Rocky Mountains is desert country.
Right, exactly.
And so how come this is rainforest?
Because the winds are bringing in moisture from the Atlantic into South America. There's been interesting paleo-ecological studies. When that convergence zone moves up and down, you can have wet, warm out west and dry in the east. And then when it moves, it just switches over. And you can have wet in the east and dry in the west. And that's not been understood in the past. And so understanding the paleo history, but there's a lot of air coming in from the south, running up and hitting the mountains and then rising up and dumping a lot of water.
Okay.
And the rain shadow is on the other side because of that.
Okay,
Yeah.
thank you. And the other question was, early on in the talk, you talked about these regions which separate the species of the bird into two subspecies, I guess, being defined by a river, as if the river caused that. Now, how does a river stop a species which flies?
Well, okay, I'm glad you asked that. You know, people think birds fly all over. Most birds actually don't fly all over. Most bird species are narrowly endemic. And even strong flyers don't go across. So I can give you I got two independent examples of this from two different people in Amazonia. Toucans, you know, the big-billed toucans you see in zoos, they look like they're pretty strong flyers, you know, they fly out in flocks. On two separate instances, people have seen individual and a flock start flying over the Rio Negro and drop in. They could not make it across the Rio Negro because even though they look like they're strong flyers, they are very narrowly endemic in a lot of those areas too. So these barriers are they are physical barriers to birds that do not fly very far. So most of the understory birds are not big, strong flyers. They live in the understory, and so even small rivers isolate some genetically.
Towards the end of your talk, it made it sound as if the river, rather than causing the speciation, defined two different geological areas which cause the speciation.
Yeah,
in other words, if you rise here and you don't here, you might get a river in between, and it's really the change in climate, for example, in the rising area which causes the speciation.
There's been a long debate within ornithology about what were the barriers that created all the species diversity. Some years ago, there was the idea that the Pleistocene climate drove the rainforest in cold times back into small refuges. And things were isolated there by ecological conditions between these forest refuges. And I have another slide that shows that our group of birds falsify that because they are older than what these refuges could have possibly been. And then actually geophysically and climatologically, those refuges don't seem to have been happening in Amazonia very much. There's no evidence, physical evidence for them. Now, the other thing is that people have had a hard time, and I've had a hard time when I was writing these papers 20 years ago on this history, of understanding how could these big rivers be the vicarious event that separated these things. But back then, I didn't have the appreciation of how rapid the changes of these rivers are in time. Because these birds, 100,000 years, 200, 300,000 years, you can change rivers right and left. And there have been some studies, not documented as well as they need to be, that there were major reductions in rainfall into Amazonia for, maybe a couple thousand of years, and that would have changed the landscape. If you reduce the rainfall coming into Amazonia, then all the headwaters become land, and birds and other things can move across the headwaters, and that's been a long postulated idea. But with climatology evidence, maybe we can begin to actually show whether that is true or not. But you can imagine, in just hundreds of years, if you take away a lot of rainfall, as some of these climatic events can do, then you could have rainforests grow across where those river systems are, and all of a sudden the rain comes back and those river systems are reestablished. So it's a very dynamic landscape which we want to try to get a hold of. But rivers can move all over, and if they do, they can divide up ranges of birds, ranges of mammals, fishes get caught into different river systems. So personally, I do think it is the river systems with climate change changing vegetation types in different places. But we don't know where those are right now, but we're absolutely looking at it because we know there's evidence for drying in certain areas, but how extensive was it? Did it split things apart? Yeah.
Let's give it on that side the question.
Okay. I'm Dave Rabinowitz. I'm a member. And I was wondering,
I'm Joel Cracraft. I'm a member now. That's
great.
great.
Okay.
Feels good.
Yeah. Your work has all been done with basically one genus of birds, I guess. And, or close like that.
This study, yes.
Yes. I'm just wondering, are you working with any other groups that are looking at correlating your discoveries with plants, insects, and other vertebrates?
Yeah. I actually should have said that, and I apologize. So from the systematic evolutionary point of view, we're looking at virtually all birds in Amazonia, all different groups of birds that at least have major endemics and major distributions. We're also looking at all primates, because primates are in the same areas of endemism as the birds. So we're doing dense phylogenetic analysis of those. We're looking at two groups of heliconius butterflies. So these are groups that have been surveyed a lot, and we're doing phylogenetics of butterflies, and then we're looking at two major families of trees, because we know that these birds and butterflies and trees don't necessarily have the same areas of endemism, and they don't necessarily have the same history. And so that way we can layer history using these. But we're doing Bignoniaceae, these lianas, and then we're doing tree tree of the Lecidacea, so but the Brazil nut family, all the species in those two families, and there's several hundred, 200 or 300 species in those families. So we are trying to look at it, and we're hoping that we will get people saying, oh, I'm working on these groups, and can I contribute? You know, we don't have money to support research, but we hope to build up more and more of a network of other people doing that kind of thing.
Carl.
Hi, I'm Carl Merrill, a member of the Society. There's no question that geophysical aspects play a major role, but one thing you didn't touch on was the biological competition. So you could have a species come in and wipe out a whole species like we're seeing in the Pacific now with the snakes that are being brought in by the airplanes and wiping out all the birds on a particular island. And viruses do the same thing. There are certain primates that have virus sequences in their genome that we just don't see in others. So that can play a major role, which might be independent of the geophysical effects. And maybe by looking at all different species and looking for correlations, you could perhaps show which is which. But I wonder if you could address that.
There's no question that biotic interactions can affect distributions of organisms. And it's just the scale of our project. We can't do population level ecology without another team of 17 institutions and do it across many, 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 many species in an area. So, I mean, we're not discounting that. We are frankly trying to look at the historical signature. Now, one of the nice things about getting the historical signature is that things that don't match those historical signals, and there's not going to be one signal, there's going to be dozens of signals because we're dealing with deep time here, relatively deep time, but things that don't match those. So if you get a whole bunch of species that are sympatric with one another in the same genus, then if you can't tease those histories apart, then maybe that is an instances of where you need to be looking at, and to explain those distributions at population ecology. But we're looking at landscape ecology. We're interested more in the big picture of where is diversity distributed. And you'd be surprised how little we actually know about that. We have bird watchers looking at birds, but you need physical documentation of specimens in a particular georeference framework to really address that kind of stuff quantitatively in Amazonia. We don't know where a lot of birds are distributed in their entirety. You know, it's a vast, vast area. So hopefully we'll move toward that. But I don't disagree with that at all, you know, that there are other things that might affect this.
Okay, Mr. Rosen.
David Rosen, a lifetime member of the Society. What would be the signature or of sympatric speciation, and how would you tell? And are you sure it isn't important?
In birds, it's as far as we can understand, and invertebrates in general, it's not important. There are a few exceptions to be sure, but in birds, it's not important. Allopatry is very much important. To document sympatric speciation, I've got slides from my evolution class that I could show. It's a very complicated thing. First, if you're postulating speciation, those two things better be sister species to begin with, right? And even with sympatry, you've got to find a way that the gene pools get separated in some way. And they may be in the same area, so you could have a host-specific plant on one species, and somehow or other there's a dispersal to another host-specific plant, and they become genetically linked to those two different plants. And that's one way it could happen, but that's not really in any of the groups we're dealing with, I think. Even the plants, are allopatric, you know, in their... Not in our groups. It would certainly be something you would think about if you were looking at insects. The butterflies don't seem to show that. The butterflies are also endemic in areas, but usually they're smaller areas than the birds and primates. But the sister taxa tend to be allopatric in this group of heliconius butterflies. In other groups, you get these really weird complexes of species for which the relationships are now starting to get worked out. And there's all kinds of genomic things going on with mimicry patterns. But again, you always have to get with few exceptions, things like polyploidy in some plants, where a plant right here can duplicate its genome and not cross back with its parental types. But that doesn't happen in birds. You know, it's just not something that's ever been observed in birds or primates, to my knowledge. Plants, most of my botanical colleagues, they see a lot of sympatry within species of a genus, but they're dealing with things that are older than these primates and birds, and so they've had plenty of time to kind of move around on the landscape with climate, with vegetation change. But sympatric speciation, I think universally people don't see it as important overall as allopatric.
Any other questions?
Hi, I'm Sarah Swanky. I'm not a member. I was wondering how deforestation and regulations have affected sampling for the birds especially. Has that impacted the ways you're able to sample?
Well, what has affected the sampling is not so much the deforestation, although it has in recent years affected it. What affects sampling of almost all organisms in Amazonia are roads. So if you plot birds, you just see all these collecting localities along roads or near roads or along rivers where you can go down a river and then get off and sample. But now that they're building roads in many places, that will open up areas for sampling, but they'll be along roads. But deforestation, it's 20%, and it's mostly in many areas to the south where they're clearing for cattle ranching. But it's a big problem. But you know, I'm sitting at the American Museum, and Terry there is sitting at the U.S. National Museum, and we're sitting on the biggest genetic repositories in the world here, and we can sample the genetics of things that are very, very rare now because they're in collections, and that's getting better and better and better. But I don't think, I think the thing we need to worry about more is climate change, because on a temporal scale, that's going to cause a tremendous amount of problems everywhere, and a lot of Amazonia it looks like it's going to disappear from climate change. You'd have to build a gigantic amount of roads over the next 50 years to do as much damage, I think, as climate change. We are not specifically doing applied research. That's not part of the program of these funders. But we think that our results will have implications for understanding flows better than we do now, historically anyway, coming to the present. But it's a good question.
One last question. Maybe go ahead, Tom.
I'm Tom Muller, a member of the Society. Thank you. You mentioned briefly the fossil record, but it's my understanding that in the Amazon area there are very few fossils because of the wetlands problem. Could you comment briefly on the fossil record?
All right. Well, the fossil record for birds is not great. There are a few fossils. The fossil record for mammals is pretty good in places out west. So you get out toward the Andes, there is a fairly well-known fossil record out there, Miocene fossil record, and a few older. But as you're absolutely right, you know, most of these fossils that are laid down, the deposits are washed away by water, and it's a terrible environment to try to do paleontology in. But people from our institute and one of our collaborators has done a lot of paleontology work on mammals out west. So there are places where we can get a pretty good idea of the mammals, but birds, no. What I was alluding to is we can build these trees and go deeper in the bird tree and find groups that have fossil record and use those to calibrate the whole bird tree. And that's what we also are trying to do and refine that estimate.
And last question, the gentleman in the back.
I am. Hi, can you? Okay. I'm Arslan Danish. Do you have any simulation tool that you use studying evolution or whatever you're doing right now?
Simulation?
Simulation tool?
The only, I think, simulation that we're planning right now is simulating climate and is simulating forest cover using global climate models and then vegetation models. That's I think right now that's what we're going to do. Now you do some simulation in genetic analysis, but I think maybe it's not what you are sort of referring to. Tools?
What simulation tool you?
Well, I'm not a climate modeler or geophysicist, but there are general climate models. There are various climate models that can be used. Oh, for genetics? Well, you can simulate populations to see whether what your pattern you're seeing is non-random or not. Those kinds of simulations. But we're not doing hardcore population genetics. We're trying to get an idea of the genetic structure and the history of that genetic structure. And in some instances, maybe demographic history of expanding populations or stable populations.
Well, thank you very much.
Thank you.
Besides members, we have one more present for you. This is the announcement signed by the members of the society, and another benefit for giving us speeches
Thank you
this
so
evening.
much.
Thank you very much.
Thank you very much. Great question.